Hello, my name is Carla Saleh, and I am a head of a unit that studies antiviral immunity in insects. So why are insects efficient vectors? As you know, insects like mosquitoes transmit several diseases to humans. Today we will specifically talk about viruses and viruses transmitted from mosquitoes to humans. So what makes mosquitoes special? Why are mosquitoes transmitting all these viruses? The answer to that is that mosquitoes are capable of developing what we call persistent infection. What is a persistent infection? And please don't mistake this with chronic infection in mammals. It's very different. In insects, persistent infections are characterized for the abundant production of viral progeny that is maintained in the time without loss of fitness. That means that the mosquito is full of virus, however, it is behaving very well. It is not sick. This is in big contrast with what we call acute infections. In acute infections, we have a peak of production of virus, and that normally will be resolved by the death of the infected animal or by the clearance of the virus. So what is unique about persistent infection is that we have a perfect equilibrium between the viral infection and the immune mechanism of the host to defend against this virus. And why is persistence so important in order to transmit virus? Because it has been proposed that the persistence is key for the transmission and dissemination. You have this mosquito that is full of virus, however, is happy, is not sick, so it can fly, it can find you, it can bite you, and transmit the virus. So one way of interrupting transmission from mosquito to humans will be breaking persistent infections. But in order to break persistent infections, it's important first to understand how persistent infections are maintained and established. Insects, like all of us, has an immune mechanism. Insects have only innate immunity. They don't have adaptive immunity. The innate immunity of insects is exquisite, with several pathways to defend them against different pathogens, bacteria, fungi, and also viruses. The pathway that defends insects against viruses is called RNAi, where I is for interference, the RNA interference pathway. How this pathway defends insects against viruses? When a virus, an RNA virus, infects the cell, we will have the viral RNA. The virus will replicate, that means that will create replication intermediates of double-strand RNA. This double-strand RNA is always a danger signal that is telling to the cell that there is a virus infection going on. So this double-strand RNA will be detected by an enzyme called dicer that will dice it in small fragments called siRNAs. These siRNAs will be then loaded into a complex called RISC with a protein called Argonaute that is the effector, and they will scan the cytoplasm searching for RNA that is completely homologous in sequence to the little one. So the siRNA will be completely homologous to the viral RNA. So RISC with the small RNA will target the viral RNA and cut it. And in this way, RNAi will be working as a hammer that is breaking the viral genome, therefore hindering viral replication. That is a very efficient system. However, we know that RNAi alone is not sufficient to reach persistent infection. You can have a totally functional RNAi response, but still go into an acute infection if you are an insect. So something else is lacking. How we reach persistent infections. Some years ago, we discovered an incredible twist to the RNAi pathway. We found that at the same time that the RNA pathway is working, viral RNA will be interacted with endogenous retrotransposons. What are retrotransposons? Retrotransposons are a type of transposons. Transposons are mobile genetic elements that are present and are very abundant in the mosquito genome. So retrotransposons have a protein called reverse transcriptase that what it does is to copy RNA into DNA. That is part of the cycle of the retrotransposon. But in this case, the virus will interact with the retrotransposon, and the RT will copy the viral RNA to produce viral DNA. So now we have fragments that can be linear or circular, So now we have fragments that can be circular or linear of viral DNA. This viral DNA will generate transcripts, and these transcripts will generate new siRNAs. So what we have now? A boost of the immune response because we have more abundant and more diverse siRNAs that will enter into the RNAi machinery. So what is persistence? It's this metastable equilibrium that you can obtain between the trigger of the immunity and the boost of the immunity due to the production and the synthesis of viral DNA. So since the discovery of viral DNA by our lab, several other teams have discovered viral DNA for many RNA viruses in different invertebrates. Today, we know that viruses produce viral DNA in flies, in mosquitoes, in ticks, in the shrimp, and also in Bombyx mori, or the silkworm. So it seems that an invertebrate is a theme, and in order to reach persistent infection, that is the equilibrium between viral infection and the immune mechanism, in one side of the seesaw you will have viral replication, and in the other you will have the immune response, the RNAi interference, plus the viral DNA pathway. Is that important in the mosquito? Is that happening in vivo in a mosquito? So yes, this is an experiment that showed that persistence relies completely on viral DNA production. So in the lab, we have mosquitoes, and we treat these mosquitoes with ACT. ACT is a base analog and is an inhibitor of reverse transcriptase activity. So we treat mosquitoes with sucrose, that is how normally they are fed, or with ACT, and then we offer them a blood milk containing chikungunya virus. We expect that the mosquito that have eat ACT, it will not be capable of producing viral DNA from chikungunya virus. And here you can see a survival curve where mosquitoes that eat ACT, they are okay, they are not affected by the drug. Mosquitoes that are infected with chikungunya alone, they survive because they are persistently infected. However, mosquitoes that are fed with chikungunya and ACT, they die. And they die without affecting the viral titers. So that is showing that if I preclude the mosquito for the ability to synthesize viral DNA, I can render the mosquito intolerant to the viral infection. Now the mosquito is not persistently infected anymore. And why this is important? Because that is demonstrated that I can selectively eliminate infected mosquitoes for a population of mosquitoes. And it's showing that I can finally move my persistently infected mosquitoes from this persistent stage to a lethal stage by treating with this drug, ACT. And when I move my mosquito from persistence to lethal, I am stopping transmission. So what we have learned today? We have learned that invertebrates that are infected with RNA viruses produce a viral DNA form. This process is called endogenization. Endogenized viral sequences participate in the antiviral immunity. The viral DNA is required for the survival of the infecting animal. If I don't have viral DNA, I cannot reach a persistent infection. And finally, we have shown that retrotransposons actively participate in antiviral immunity. What is really important, because retrotransposons are highly abundant and we don't know exactly what they are doing inside the cell. And now we know that this part of the genome is participated in innate immunity. Even more important, what we are showing here is that insects that only have innate immunity have found a way to amplify and transmit information in the immune status by using nucleic acids. So you could do a parallel. In mammals, you will use antibodies, but insects do not have adaptive immunity. So inside the innate immunity, they have found ways to transmit information in an infectious state using RNA and DNA. Thank you very much for your attention.